Hi, this is Mrs. Murdock, and I'm going to talk today about photosynthesis. So, this is the process of photosynthesis at the AP biology level. AP photosynthesis. Okay. Synthesis photo, meaning to make synthesis with light photo, right? Okay. So, I'm only going to get through to the light reactions today. There's two processes, two parts of photosynthesis. The first are the light reactions, and the second is the Calvin cycle. Just going to do the light reactions here, but I'm going to put it into context first. So, photosynthesis happens in the leaves of plants, in the photosynthetic tissue of plants. You're not going to find it in the bark or like in a potato or in the fruit. You only find it in the leaves, which are the organs that actually have chloroplasts in them. So, picture you've got a leaf cell, right? And then within the leaf cell, you have a chloroplast. And I'm just going to draw a big not in red though. Yeah, let's stick with black. I don't know why it does that. Oh, come on. There. Let's just keep everything neutral for now. All right, so if we draw a big chloroplast here, all right, so remember, out here is the plant cells cytoplasm out here, right? And we might have mitochondria next door. Don't forget that, because plant cells do both. They do both respiration and photosynthesis. But we have a chloroplast here. And there's two main structures inside that you really need to know. One of them is thylakoids. These thylakoids that make these stacks that almost look like stacks of pancakes. Some people call them stacks of coins. And these are called thylakoids. And out here, there's a bunch of sort of syrupy, enzyme-filled liquid called the stroma. It's called the stroma, which is the liquid, right? Liquid. And the thylakoid membrane, this is where the light reactions happen. The light reactions happen here, which are the first step of photosynthesis. They happen in and around these thylakoid membranes right there. And then the second part of photosynthesis is called the Calvin cycle, and it happens out here in the stroma. The Calvin cycle happens out here in the stroma. We sometimes call these the light-dependent reactions. Nope, that's not right. Light-independent reactions. Because they don't need light in order to happen. They don't directly need light. But they do need the products of the light reactions in order to happen. So you have to have the light reactions happen first before the Calvin cycle and the light-independent reactions are going to be able to happen, right? So those are the two steps. There's a worldview, a pigeon's eye view of photosynthesis. It happens inside a chloroplast. The first steps happen in these structures called thylakoids, which is where the chlorophyll is, right? So I'll make these green because that's where the chlorophyll is, and that's the ingredient that helps the light reactions to work, right? That's the first steps. And then the second step is called the Calvin cycle. It happens in a series of enzyme steps out here in the stroma. All right. So now that we've kind of located things for you in terms of helping you figure out where things are happening, let's just concentrate on the light reactions only. So what I'm going to do is I'm going to draw a picture that's just this piece right here, okay? This one piece of the thylakoid membrane. And that's where the light reactions happen, and that's what we're going to do. Okay, so this is the first step of photosynthesis, often called the light-dependent reactions. Dependent reactions. Okay. And we're inside the chloroplast, and we're inside the thylakoid membrane, or the thylakoid membrane is here, right? Just kind of draw it like this, thylakoid membrane. And if this is the thylakoid membrane, then out here is the stroma. right? The liquid stroma is out here, and inside here is, let's call it inside the thylakoid, is inside here. Okay. So remember that this is a little bit similar to the mitochondria. There's a double membrane in a chloroplast, just like there is in a mitochondria, right? So you've got an inner chamber. The inside the thylakoid, those stacks of coins each have an inner compartment. And then out here you have the stroma, right? Which is where the Calvin cycle is going to happen. And the Calvin cycle is waiting for, right? Waiting for the ingredients that are going to come out of the light reaction. So let's talk about that. So the first thing that you need to know is that there are chlorophyll molecules embedded in the membranes, right? And often you'll see in your textbook, you should have seen them called photosystems, and there's two of them. There's two photosystems involved in photosynthesis, and they're both bundles of chlorophyll molecules. And the reason why I'm drawing these funny sort of flowers with stems is because a chlorophyll molecule looks a lot like that. A chlorophyll molecule has this sun-catching array of carbons and magnesiums up on the top, like this kind of like a circular thing. And then it has a long stem that's made of the same thing that those phospholipid tails are made of, hydrocarbons. It's a long hydrocarbon chain that helps it anchor itself in these phospholipids that are all around it. So structure and function, right? Here's the sun-catching part of the chlorophyll, right? These are oh, I better not do that because that won't give me room. I'll do that on the other side. Okay. So these photosystems are bundles of chlorophyll molecules, right? The chlorophylls are kind of in bundles in these photosystems. This is PS2 and this is PS1. Photosystem 2 is used first, photosystem 1 is used second. I know that doesn't make sense, but it has to do with the order in which they were discovered, right? So they were numbered that way, even though it kind of seems backward to us. So those photosystems are bundles, bouquets of chlorophyll molecules that are ready to catch sunlight and convert its light energy into chemical energy. So here comes the sunlight, right? Sunlight comes along. And the first thing that happens is you have these electrons that are in the chlorophyll, the center of the chlorophyll molecules that get excited. So again, we're doing a flow of electrons here. And the electrons, just like in the mitochondria, they're going to flow through an electron transport chain. So that's what these guys are. We have an ETC here, just like in the mitochondria. And the electron, once it gets excited, it's going to flow down through these ETC proteins, right? Just like it did in the mitochondria. Same type thing. And just as happened in the mitochondria, these special ETC proteins have the ability to pump protons. So hydrogen ions are once again going to be pumped across a membrane as a result of electron flow. And we're going to build up a proton motive force, just like we did in the mitochondria. So there'll be a whole bunch of hydrogen ions in here. That thylakoid space fills up with protons. Okay. All right. And just like with mitochondria, we also have a ATP synthase here. Another ATP synthase molecule that when hydrogen flows through it, which it will because hydrogen likes to get rid of that imbalance. Remember, nature doesn't like an imbalance. It doesn't like to have more hydrogens in here than there are out there. The only opening is the ATP synthase. And we are therefore making ATP molecules. Okay. So that solar energy is being converted into the potential energy of ATP. There's your first useful thing that the plant is doing. Okay, that's an important conversion. Go ahead and make that red. Okay. There. Right. There. Right. That's the first thing that we get out of the light dependent reactions. Now, here's a problem though. These electrons that are leaving these chlorophylls, they leave behind a hole, right? There's a hole here where the electrons are. And nature doesn't like that either. We need to fill that hole. So what happens there is you have water that's going to be split. Water is split. That's how you make oxygen. That's also how you make some hydrogen ions, but you're making oxygen and breaking those bonds to create electrons that go to fill that hole that was created. Right? So that process there is called photolysis. You are splitting water, and those electrons go to replace the ones that were lost. So the origin of the oxygen that comes from photosynthesis happens at the very beginning of the process. Whereas if you remember the mitochondria, that's the last thing, right? Oxygen comes in the last thing in cellular respiration. In photosynthesis, it's the first thing. Oxygen is consumed, is produced right away, right? Splitting of water, you have oxygen. Okay. All right, so that's the first part of this. Now we have this other photosystem, and the electrons have landed here, right? These electrons have landed here, and now they're sitting there waiting for what happens next. What happens next is the sun hits it and gets them re-excited. So we're going to re-excite those electrons. They're going to flow through a very short ETC again. But they're going to land on this protein right here, which instead of pumping protons, is going to allow that electron to reduce NADP+ into NADPH. There. There. Okay. So we've just used those electrons, those high energy electrons, right? The electron that came up, that got excited by the light, to reduce this guy, which is the other thing that now needs to go to the Calvin cycle. The ATP goes to the Calvin cycle. The NADPH, which is an electron carrier just for photosynthesis, easy to remember because there's a P in it for photosynthesis. Isn't that nice? Okay. And those are the products of the light-dependent reactions that the Calvin cycle is going to need to make its sugars. And that's the light-dependent reactions. The next step is the Calvin cycle, and we'll talk about that next, and I hope that was helpful.